The antioxidant story of creatine is one of the more surprising dimensions of this molecule, and one of the least discussed outside of specialist research circles. Creatine is not a conventional antioxidant in the way that vitamin C or vitamin E are. It doesn't directly neutralize free radicals through electron donation, but its interaction with reactive oxygen species production during intense exercise is real, indirect, and meaningful enough to show up consistently in the research. Here's the mechanism. During intense exercise, your muscle cells produce reactive oxygen species, often abbreviated ROSI, as a byproduct of the rapid mitochondrial ATP production and the various enzymatic reactions happening under high-demand conditions. A modest amount of ROS is actually necessary for normal exercise adaptation. They serve as signaling molecules that trigger mitochondrial biogenesis, antioxidant enzyme upregulation, and other adaptive responses. But excessive ROS production during very intense or prolonged exercise can overwhelm the cell's antioxidant defense systems, causing oxidative damage to proteins, lipids, and DNA within the muscle cell. This oxidative damage is one contributor to exercise-induced muscle damage and the inflammatory response that follows. Creatine supplementation affects this dynamic in two ways. First, by maintaining a larger phosphocreatine reserve, creatine allows muscles to sustain ATP levels more effectively during intense effort. When ATP levels drop sharply, as they do in unsupplemented muscles during maximal effort, several enzymes that are ATP-dependent and involved in antioxidant defense begin to lose function. By preventing deeper ATP drops, creatine indirectly preserves the function of these ATP-dependent antioxidant systems. Second, there is direct evidence that creatine interacts with certain reactive oxygen species. In vitro studies in isolated cell systems have shown that creatine can directly scavenge some reactive oxygen species, particularly peroxynitrite, a reactive nitrogen species formed when nitric oxide and superoxide combine. Peroxynitrite is one of the more damaging reactive species in biological tissue, capable of nitrating proteins and causing lipid peroxidation. Creatine's ability to scavenge peroxynitrite in cell culture systems doesn't necessarily translate to the same magnitude of effect in vivo, but it does suggest a direct chemical interaction that goes beyond the purely indirect ATP-mediated mechanism. In animal studies, creatine supplementation reduces markers of oxidative damage in muscle tissue after intense exercise. Supplemented animals show lower lipid peroxidation products, lower protein carbonylation markers, and preserved antioxidant enzyme activity compared to unsupplemented controls. Human exercise studies show similar patterns: lower post-exercise oxidative damage markers in supplemented individuals, consistent with a combination of ATP-mediated antioxidant preservation and possible direct ROS scavenging. The practical implications are most relevant for athletes whose training involves very high volumes of intense exercise, where ROS production is substantial enough that oxidative damage is a meaningful contributor to fatigue, soreness, and recovery time. For moderate volume recreational training, the antioxidant dimension of creatine is a secondary benefit. For elite athletes training twice daily, the oxidative stress reduction may contribute meaningfully to recovery capacity and long-term cellular health. The clinical picture is more compelling when you look at specific disease states. In conditions involving mitochondrial dysfunction, including certain muscular dystrophies, Parkinson's disease, and traumatic brain injury, oxidative damage is a central mechanism of disease progression. Creatine supplementation has been studied as a neuroprotective and cytoprotective agent in these conditions, partly because of its ROS modulating properties. The results in Parkinson's disease research are mixed. Some studies show modest benefits for symptom progression, others don't, but the mechanistic rationale is sound. In traumatic brain injury models, creatine given before or immediately after injury consistently reduces the degree of oxidative damage and improves functional outcomes in animal studies. Human trials in TBI are ongoing. For healthy athletes, the most relevant practical implication of creatine's antioxidant properties is in training blocks involving very high eccentric loads. hill running, heavy Romanian deadlifts, intensive plyometric training, where ROS production is substantial. Creatine's indirect antioxidant effect through ATP preservation, combined with potential direct ROS scavenging, contributes to the lower post-exercise muscle damage markers seen in supplemented athletes in exactly these high eccentric load conditions. That's not the primary reason to supplement.